This is Epic Book Recaps, a podcast that offers complimentary audio summaries of influential nonfiction books, making learning a smarter experience. The Gene by Siddhartha Mukherjee. The roots of genetic research were shrouded in a myriad of confusion, misconceptions, and theories, yet these early conjectures about the concept played a pivotal role in molding the future of genetic studies. Historically, it was believed that the male parent was solely responsible for the genetic data of the offspring, a notion that was commonly accepted since the time of Pythagoras around 530 BC, in a theory known as spermism. This theory proposed that men provided the sperm and the woman's role was merely to offer a nurturing environment and sufficient nutrition for the fetus to develop in her womb. This concept formed the earliest understanding of heredity and inherited traits, which were key aspects in the study of genetics. The belief emerged that inherited genes were based on the triangle theorem, which was perceived as a blueprint for perfect pairing, and that it was possible to manipulate this law of birth. This conclusion was credited to Plato, and it would have held sway among the intellectual elite if it had not been for Aristotle's counterargument. Aristotle posited that an offspring could inherit traits from both his parents and their parents as well, and not just their fathers and grandfathers. Aristotle continued to dispute spermism by citing various examples where certain predominant traits were inherited from the mother's side and the father's traits skipped a generation. He ultimately argued that the notion that the man alone was responsible for the creation of a child was flawed since a female cannot be formed with the genetic data of a man. He postulated that genetic matter was contributed by both parents, and that was what formed the finished product in the form of information. This sparked a myriad of theories on the subject until one scientist was able to interpret the code for all this and develop a theory to fully explain heredity. Charles Darwin, through his numerous journeys, inquisitive mind, and thorough research, determined that organisms propagated their own essence by sharing genetic material, and that these traits were passed down or faded away, depending on what he termed as natural selection. His theory revolutionized the approach to the issue of heredity from that point onwards. He argued that each species is a descendant of a much older variant, one that adapted itself through birth to continue to exist but eventually died off. The origin and survival of a species are greatly influenced by its genes and their physical interaction with the environmental triggers around it. Scientist George Mendel argued that as species mate and prepare their offspring for survival through evolution, certain traits from both parents become either dominant or recessive. This implies that in order to survive, certain traits will be relegated to the background, while others will be noticeable for generations. Essentially, the theories of Pythagoras, Aristotle, Plato, and all the other great minds before had simply funneled into one solid conclusion. For a species to extend its existence, it must first share traits with the opposite sex during mating, and then must promote only the features that would be instrumental in its survival while inhibiting the ones that would endanger it. This revelation took a dark turn as it paved the way for more critical thinkers to explore a theory known as eugenics. This theory entails eliminating the weaker variants of the human species and promoting the superior ones. According to Francis Galton, a relative of Darwin, this was the natural progression of the species, and he was merely expediting the process. This theory would fuel numerous terrible events throughout history, including the Holocaust. Further studies on the gene revealed that it was located on the determinant substance for fetal development, the chromosome, and occurred in genomes. The question had now shifted from how species propagated their genes, or how these genes evolved to suit survival, to the question of what a gene actually is. Several theories had emerged about genes and inherited traits, but nobody knew what a gene really was, or how it really affected us as a species. Mendel had been able to state clearly that heredity worked in units, small portions of a species propagating all the useful information about it and replicating it along the line. He called this a gene. Although Mendel discovered the gene, it was William Bateson, a scientist, who was first able to unravel the mysteries of the gene. However, even with Bateson's work, understanding of the gene eluded us until a German scientist, Hugo de Vries, delved deeper into the mysteries. It would fall upon Thomas Morgan, a zoologist, to further expand upon the groundwork laid by the people before him. He observed the gene and was able to adequately explain how it was found in chromosomes, and that the traits of a species were largely dependent on the chromosomes. The gender of the offspring, height, and every single noticeable feature was determined by the chromosomes carrying the genes. Genes weren't only responsible for the transfer of traits across generations. It would later be discovered that genes were in fact responsible for much more. The gene contained data, information critical for the entire body to function, and directed how it should function. It controlled mating patterns, metabolism, allergies to certain foods were also encoded within the gene, etc. The studies on the gene were quite delayed because no one appreciated its minuscule presence but pondered about its major impact. As the curiosity around the workings of the gene grew, so did the need to understand how it governed the formation of organisms. The field of embryology was energized, and it would pave the way for a deeper understanding of what was once simply ignored by the world of science. Genes weren't indivisible materials. They had a code, a sequence that orchestrated the formation and transformation from embryo to fetus to full-grown organism. Heredity operated on a code, a genetic framework that also relied on a series of commands or messages that were embedded in the genes of each and every creature, and this code could be used to alter certain traits to favor survival. Like amending cancer genes or susceptibility to other illnesses such as diabetes. Genes were found to be responsible for the unexplainable illnesses that plagued humanity, and upon research, scientists were able to isolate and identify these genes. Gene alteration was seen as the future of modern medicine and a solution to whatever incurable illnesses we had to deal with. The concept of genetic alteration didn't slow down as time progressed. Instead, it became an obsession. The arguments put forward by ancient philosophers about cracking the code of inherited traits and grooming the human race to only consist of elite members had gained momentum. But now, it wasn't just theoretical, it was a reality, and the world had begun making significant strides in both research and experimentation to produce results. By the 1970s, due to earlier discoveries on gene transformation, scientists had the idea of altering DNA by introducing foreign genetic material into the cellular unit. The earliest attempt at gene alteration was through a viral medium. A virus is simply a protein case carrying a parcel and can be repurposed to carry genetic material instead, and of course, replicate this new information on a cellular scale, thereby affecting the whole in the process. Due to its natural design, a virus is like a package delivery system that scientists can attempt to implant foreign genetic material into a human cell using a virus. But the risks of using a virus to deliver a payload that could be replicated in such a manner were too high. The virus itself could take hold and replicate alongside. This prompted further work to be done on the subject of gene alteration, and it was finally agreed that using bacterial media was a much safer alternative since they were less likely to hijack the payload and cause irreparable damage to the cells of the recipient organism. Gene sequencing is the study of how the creation, propagation, and replication of DNA occur at the cellular level. The process of DNA replication needed to be mapped if there was going to be any progress in understanding how to trace the partitions that might be ultimately detrimental or beneficial to the organism. Once this process was amply observed, it was possible to effectively clone the DNA, rewriting it so that whatever flaws existed could be corrected for the next generation. This process required DNA molecules formed in the laboratory. These molecules are known as recombinant DNA molecules. They are a construct made up of the DNA of other species to form a new, more sustainable, albeit experimental version. This process is quite beneficial in the field of medicine, especially in cases where replicating or implanting needed genes can fight certain illnesses. For example, the case of diabetes and the lack of insulin in the body can be fixed if the gene responsible for its production was replicated or implanted, and so on. Studies have shown that we are predisposed to certain illnesses by virtue of our genetic framework. The extensive scope of factors dependent on the genes of an individual means that to ignore genetics would lead to certain extinction for a species. For instance, certain illnesses are propagated or inherited via genes. The son of a diabetic or dementia sufferer will most likely have the same fate. Although susceptibility does not mean certainty, oftentimes the illnesses require a trigger to manifest in the bodies of the heirs. To truly study the workings of human genetics, we must first understand humans and how we interact with our genetic framework. Most importantly, our interaction with diseases that stem from our genes. Examples of such interactions include the case of inherited hemophilia among European royals and the predisposition to sickle cell anemia in African and Caribbean families. Certain diseases are predominant in certain gene types, and certain people will continue to produce offspring who will suffer the same fate. These illnesses could either be caused by a single gene, monogenic, or a group of genes, polygenic. Polygenic diseases have two forms. They are either a group of genes responsible for the illness or single genes scattered along the genome. Monogenic and polygenic illnesses based on a group of genes usually result in rare and/or strange conditions such as Down syndrome and the likes, but the polygenic illness that is caused by scattered genes on the genome results in much more constant and dire conditions. Some of the conditions that may be suffered with a polygenic illness include infertility, obesity, cancer, etc., and are particularly difficult to treat or cure. If the specific genome responsible for terminal illnesses or incurable conditions can be isolated and removed, there is hope for treatment and even cures. These genetic malformations were termed mutations, and they were vigorously studied in hopes of finding a way to deal with them or manage their effects. It's worth noting that a mutation in a genome does not immediately translate to a rare disease or a malformation in phenotype. While certain mutations cause brittle bones or weak ventricles, others simply result in the ability to curl your tongue or taste PCP. But the fear remained nonetheless, and led many parents seeking prenatal testing to know if their children would turn out fine or at least normal. A rise in abortions followed as parents found out their babies weren't developing right due to a mutation. As a result, the discoveries of mutations gave rise to a new form of eugenics, where only elite individuals with desirable genes were to be bred moving forward. This new form of eugenics, nugenics, was not as savage as its predecessor and was implemented at sperm banks. This meant they only accepted donations from those with these elite genes. But with more studies came more and more complexities in the gene puzzle. Since the challenge of gene sequencing and mapping was solely centered around unraveling the code of inheritance and how to eradicate dire illnesses, diseases, scientists were able to understand certain conditions and pinpoint the actual gene or genome responsible for them. We are able to trace our histories by simply delving deeper into the mechanism of the gene to discover just where and when we originated. Genes, it appeared, were responsible for much more than initially considered by scientists. The scope of inquiry into the workings of the gene only accounted for pathology and disease propagation, but there remains so much more to discover. It would be found out that the origin of mankind, as initially considered by Darwin, was left shrouded in uncertainty. The prevailing belief was that there existed only three origins: white, Asian, and Negro, arguing that these three groups had three distinct ancestors that would later propagate into the current races we have today. This notion is largely unfounded, and it failed to present any proof beyond the bold claims of its originator. In reality, all humans originated from sub-Saharan Africa and migrated east, west, north into the current dispersions we have today. There was a prevalent misconception that the human race originated from three sources. This was a racist agenda suggesting that one race is superior to the other, but in truth, every race originated from one place. Another function of the gene was in the determination of gender, owing to the XY chromosomes. So, whether an infant is to be male or female, it is determined by the genes, predominantly those of its father. But as is the nature of genes, there are bound to be mutations, and these mutations often result in individuals of a certain gender by virtue of chromosome being a member of the opposite gender. This indicates females who have the presence of XY chromosomes in addition to the inherent 20. This usually results in the duality of sexual organs, but only one gender is visible upon observation. The gender of an individual is dependent on the genes, but oftentimes this can mutate and cause a clash of genders, leaving an individual with both genitals sometimes and hormones. The presence of an opposite chromosome in the genome of an individual triggers a series of conflicting reactions such as anxiety, anger, and depression. It was found that for some of these individuals born with latent duality of genders, their parents had authorized a sex adjustment surgery performed on them to give them just one gender. This procedure was ineffective in assigning gender to an individual. Once again, the role of the gene was irrefutable and immutable. Among the wonders of gene mutations is the phenomenon of twins. Two humans formed when either one fertilized egg splits into two identical eggs, or when two sperm cells fertilize two eggs at once. The former is a testament to the seamless replication of the genetic message propagated by the gene. It succeeds in creating two very similar individuals who, by all measures, are the same person. But in keeping with the mysteries of genetics, they can be so different mentally and can have different reactions to different substances they encounter. Scientists would go on to closely observe the process of embryo development and the ultimate role of the gene. This led to research into the key ingredient for the embryonic process, the stem cell, and would result later in experiments that would replicate the action of the stem cells in a lab. To truly understand the process of embryonic development, scientists focused on the stem cell, the operational cog in the mechanism, which showed great promise of answering previously unanswered questions. Moving forward, scientists are able to orchestrate the final product of gestation free of any of the usual susceptibilities to disease or any impediment to survival. The aim of stem cell research isn't just to replicate the development of a fetus, but to tailor the genetic setup of an infant from scratch to eliminate any malignant traits. With this research, scientists are able to isolate and sequence the progression of growth from a fertilized egg to a fully formed fetus. Armed with access to the intricacies of stem cell data, the distress of eugenics or abortion to eliminate weak stock is reduced to the bare minimum. The prospects are remarkable and the benefits for the human race are numerous. The gene sequence responsible for recurrent illnesses can simply be traced and rewritten or removed altogether. Children whose genes would normally predispose them to certain terminal conditions can be spared such a fate, and incurable diseases can simply be eradicated before they even take root. The process of adjusting predominant conditions in the genetic framework is known as gene therapy. Simply put, The presence of a protein or sequence of proteins in a gene that makes it susceptible to certain illnesses would be replaced with ones that won't. This development is revolutionary, and this might be the future that Plato arrogantly suggested would consist of only the elite or fit, as Darwin would later posit. Now, instead of abortions or eugenics, these weak or unfit people would simply undergo gene therapy and they'd be on their way. Gene therapy holds the promise of treatment and management of previously incurable conditions. Instead of laborious medication, the gene responsible for the illness can be replaced altogether. Did you know? Normalcy is the antithesis of evolution. It is quite ironic that something as minuscule as the gene is responsible for so much in our known world today, from our origins and evolution to adaptation. Everything that we see and touch in our everyday lives came from the variation of genes. Birthed from the hard work of scientists like William Bateson, Charles Darwin, and George Mendel, knowledge of the gene has grown and spread, opening new branches in biology. Modern-day science has been able to harness the crude prodding of these great minds who first took on the challenge of understanding human heredity and how it could be tailored to favor our ultimate survival, and used it to improve the quality of our medicine and life in general. It allowed us to understand our history, how our bodies work, and the possible future for us and the world around us. It has even been able to help us avoid unnecessary problems by ensuring that the next generation is healthier than the former. In the world of science today, more applications and procedures in which the gene is principal in saving lives and ensuring better living conditions for individuals have been discovered. You do not need to be a scientist to seek understanding about genes, as it can help you understand your body or help you ensure you have healthy offspring with the right partner. Do this. It is important that you identify, if present, genomes that may be responsible for any hereditary illnesses in your lineage and take appropriate therapy.